Welcome back to the deep dive. We are jumping straight into, well, one of the most frustrating, almost paradoxical challenges in computational pathology today, especially in oncology.
It really is a massive problem. And it kind of boils down to these two opposing forces, doesn't
Yeah.
it? Data scarcity versus data size.
Exactly. So on one hand, you've got these incredibly specific clinical questions, high-stakes stuff like predicting if a really rare type of tumor is going to respond to a certain drug.
Yeah, and the outcome data for those, super scarce. You might be lucky to have, what, fewer than 100 patient samples worldwide sometimes?
Right. But then the input data for just those 100 samples, it's absolutely enormous. We're talking whole slide images, WSIs, gigapixel images, just vast amounts of visual data.
Mm-hmm. And trying to train an AI model to find some tiny, subtle visual clue hidden in, a gigapixel image, a clue that correlates with a really rare outcome.
When you've only got maybe 50 examples to learn from.
It's basically impossible with traditional methods. The model just overfits like crazy immediately. It memorizes a few examples instead of learning a general pattern.
But today, we're diving into a radically different approach, something designed to actually solve this mismatch. Researchers from Brigham and Women's, Harvard, and MIT have introduced this new AI model called Threads.
Yeah, Threads. It's designed as a molecular-driven foundation model specifically for pathology. And that term foundation model is really key here.
Okay, break that down for us. What does foundation model mean in this context?
Think of it like building a universal visual dictionary for tissue. It's a massive AI, right? And it's pre-trained on just tons and tons of general data.
So it learns fundamental stuff, like what tissue generally looks like, what cancer cells often look like across many types.
Exactly. It learns these really rich, abstract representations.
Yeah.
And the beauty is these representations can then be instantly transferred to specific, much smaller clinical tasks. That's called transfer learning.
So you don't have to start from scratch building a model for every single little question.
Precisely. You leverage the foundation, but what's really fascinating and what sets Threads apart is how they build that universal dictionary.
Right. It wasn't just trained on the images alone, was it?
No, that's the core innovation. It's a multimodal approach. They pair the standard H&E stain tissue images with the corresponding deep genomic and transcriptomic profiles from that exact same piece of tissue.
Ah, so the image plus its molecular fingerprint.
Exactly. And that molecular pairing acts as this sort of unbiased guide that the model desperately needs when learning from the images.
Okay, let's dig into that guidance mechanism, because traditionally, AI models in pathology, they often tried to learn based on, say, what pathologists wrote in reports, right? Or just based on visual similarity alone.
Mm-hmm.
Why is it so critical here to kind of ignore that subjective layer and bring in the raw molecular data, the DNA and RNA profiles?
Well, that's really the heart of the issue, isn't it? A pathologist's report, even from an expert, is still an interpretation based on morphology, the shapes, the colors, the textures of the cells.
And morphology can be tricky.
It can be subjective, yeah, or ambiguous, sometimes even misleading. You could have two tumors that look, for all the world, identical under the microscope. But they might have totally different molecular pathways driving their growth. And that means they'll respond completely differently to treatment.
So the visual data is the look, but the molecular data is more like the biological ground truth.
That's a great way to put it.
Okay.
Precisely. The genomic and transcriptomic profiles give you this objective, holistic readout of the biological state. They capture clinically relevant information that you might just miss by only looking at the morphology.
Okay.
And Threads uses this molecular truth to essentially force the image model to learn visual features that actually matter biologically. It does this through a process called cross-modal contrastive learning.
Okay, cross-modal contrastive learning sounds pretty technical. Can you, give us the conceptual breakdown? What's happening there?
Sure. Imagine you've got two big piles of data. Pile A has all your WSI images, and Pile B has all the matching DNA and RNA profiles for those same tissues.
Right. Paired data.
Exactly. So when the AI is training, it takes a slide image from, say, patient X, and it also takes the molecular profile from patient X. Then the AI tries to make the visual representation it generates for that slide look mathematically more similar to patient X's molecular profile
than it does to any other patient's molecular profile, like patient Y or patient Z.
Exactly right. It's basically teaching the model, hey, the visual fingerprint you create for this slide needs to be biologically consistent with its unique DNA fingerprint. not just generally similar to other slides.
Ah, I see. So it forces the visual side to filter out maybe visually interesting stuff that isn't actually biologically relevant and focus on the features that correlate with the deep biology.
Precisely.
Right.
It learns to see what matters biologically. And to do this effectively, you need a lot of data and diverse data.
Which brings us to their training data set, MBTG 47K. That sounds massive. 47,171 paired samples.
Yep. Paired H&E slides and sequencing profiles. It's the largest data set of its kind used for this type of foundation model pre-training so far.
Where did they manage to get all that data from, and was it diverse?
The diversity was absolutely key. They pulled data from 4 major sources to ensure it wasn't skewed towards just 1 or 2 cancer types, which is a common issue.
Okay, where from?
So roughly half came from internal hospital sources, Mass General, MGH, and Brigham and Women's, BWH. That gave them high-quality clinical cohorts.
Right.
And the other half came from really crucial public databases. You've got the Cancer Genome Atlas, TCGA, which many listeners will know.
A huge resource.
And also the Genotype Tissue Expression Project, or GTX. That adds normal tissue diversity, too.
And critically, it wasn't just lung and breast cancer, right? They covered a lot of ground.
They did. The data set includes samples from 39 major human tissue sites, so they really made sure Threads learned a general understanding of human tissue, not just the most common cancers. That's a big strength compared to older models.
Okay, so quick recap of this part. Threads generates these universal WSI representations, these embeddings. But crucially, the learning process isn't guided by subjective reports, but by the objective, deep biological truth found in the NGS data across a really big and diverse data set.
That's the core idea, yes.
Now, building something like this, with that level of sophisticated pre-training, it must require some serious computational architecture. Let's talk about how Threads is actually built. It's like 3 specialized parts working together: vision, RNA, DNA.
That's right. Handling a gigapixel image is an engineering challenge in itself. So for the vision part, Threads uses a pretty advanced vision encoder. It's based on a model called CECO-NCH V1.5, which is a type of vision transformer, a VTL model.
And it doesn't try to look at the whole gigapixel slide at once, does it?
No, that's computationally infeasible.
Okay.
It breaks the image down into patches, but importantly, it uses relatively large patches, 512 by 512 pixels. That's bigger than many older models used.
And using those bigger patches helps capture more context, right? You see the cell and its surrounding neighborhood, the microenvironment.
Exactly. More morphological context. So once it processes all these patches, and then there can be hundreds of thousands per slide, it extracts features from each one. Then another part, an attention-based slide encoder, aggregates all those patch features into one single comprehensive slide embedding. That's the universal representation for the whole slide.
Okay, that covers the vision side. Now, what about the molecular data? RNA and DNA profiles are totally different structures from images.
Completely different. So the molecular branch is split to handle the 2 main types. For the transcriptomic profiles, the RNA sequencing data, which reflects gene expression levels, they needed something sophisticated.
Right, that's complex data.
It is. So they actually adapted an existing single-cell foundation model called SCGPT and repurposed it for bulk RNA-seq data. It's designed to handle those complex patterns of gene expression across thousands of genes.
Okay, so a powerful model for the RNA. What about the DNA side, the actual mutations in the genes?
For the genomic profiles, things like specific point mutations, SNVs, small insertions or deletions, indels, and copy number variations, CNVs. They used a slightly simpler but still powerful neural network. It's called a multi-layer perceptron, or MLP.
And that focuses on?
That focuses specifically on encoding the status of hundreds of key cancer driver genes. So is gene X mutated? Is gene Y deleted? That kind of information.
You got it. And the magic, again, is that the training process is constantly forcing the visual features learned by the image encoder to align with the features learned by these 2 separate molecular encoders.
That's the cross-modal learning again, yes. Constantly making sure the visual representation matches the combined molecular reality.
It's definitely a complex bit of engineering, tailored for these very different but linked data types. But okay, architecture aside, the million-dollar question is, did it work? Did all this complex, expensive pre-training actually pay off in terms of clinical utility? What did they test it against?
Well, this is where it gets really impressive. They didn't just test it on 1 or 2 tasks. They ran what is arguably the most comprehensive benchmark we've seen in computational pathology for a foundation model.
Okay. How comprehensive?
Threads was evaluated across 54 distinct oncology tasks. 54. using 23 different patient cohorts sourced from 17 different institutions. Just a massive evaluation scope.
Wow, okay. 54 tasks. What kinds of clinical questions did that cover? It must have been a huge range.
It was. They grouped them into 4 main families, sort of going from more established tasks to the really tough cutting-edge ones.
Oh, boy.
So first, you have things like clinical subtyping and grading. 8 tasks there. Think standard stuff like Gleason grading in prostate cancer or classifying lung cancer subtypes.
Okay, standard diagnostic tasks.
Then, gene mutation prediction. 21 tasks. Predicting from the image whether specific driver genes like KRAS or EGFR or IDH1 are mutated.
Which is huge for targeted therapy. What else?
Third, IHC status prediction. 12 tasks. Predicting the expression levels of important proteins like PD-L1 or HR2, usually measured by immunohistochemistry. Again, vital for treatment decisions. And finally, what many consider the holy grail, treatment response and survival prediction. 13 tasks in this group, predicting how patients will fare or if they'll respond to specific therapies. And this last group often involves the smallest, most difficult to work with patient cohorts.
The exact area where data scarcity bites hardest.
Exactly. So they threw this massive battery of tests at Threads, comparing it against other leading foundation models like PRISM and GIGAPATH.
In the results, did the multimodal pre-training actually translate into better performance, especially on those tough tasks?
It was pretty decisive. Threads came out on top. It achieved state-of-the-art performance, significantly outperforming all the baseline models across the board.
Significantly how? Like by how much?
On average, across all 54 tasks, it showed an absolute performance gain of 6.3% compared to the next best competitor, which was PRISM. That's a pretty substantial jump in this field.
Okay, 6.3% average gain is impressive. But I need to clarify something important for listeners. Did they have to take Threads and then spend ages fine-tuning it heavily for each specific task out of those 54? Or did it perform well more easily?
That's a great question, and it really highlights the power of the foundation model approach. Now, the superior performance they reported was achieved using just simple linear probing.
Linear probing, what does that mean practically?
It's the ultimate test, really. It means they took the complex universal image representation that Threads had already learned during its massive pre-training.
The embedding.
Right, the embedding. And they just put the simplest possible classifier on top of it, basically a single linear layer. No complex, deep fine-tuning for each task.
So the heavy lifting was already done. The visual features Threads learned were already so well structured and biologically relevant that you barely needed to teach it anything new for the specific task.
That's exactly it. That efficiency is successful transfer learning. For instance, on common tasks like identifying invasive ductal carcinoma in breast cancer, Threads, with just linear probing, hit 98.3% accuracy.
Which is competitive with models specifically built and fine-tuned just for that one task.
Yeah, competitive with much larger specialist models, but achieved with minimal extra effort because the foundation was so strong.
Okay, that really validates the pre-training strategy, but let's circle back to the original pain point we started with, data scarcity. Foundation models are great when you have lots of data for the downstream task too, but the promise here was helping with those small cohort high-stakes problems, like treatment response prediction.
And this is really where Threads delivered arguably its most important result. Remember those prognostic and treatment prediction tasks? The ones in that 4th family.
Yeah, the ones with tiny cohorts.
Tiny cohorts, exactly.
Cool.
Often just 36 to maybe 144 patients. Threads showed superior performance in all 7 of the specific treatment prediction tasks they tested.
All 7 examples.
Yeah, things like predicting patient response to bevacizumab in ovarian cancer or to temozolomide in glioblastoma. These are scenarios where patient numbers are notoriously limited, and finding reliable predictive biomarkers is incredibly hard.
So its robust, biologically informed foundation allowed it to generalize better from limited data where other models might struggle or fail.
That seems to be the case. It wasn't just better on average. It was particularly strong, precisely where we need AI help the most in these difficult, data-scarce scenarios involving rare diseases or prognosis.
That's really compelling.
And this efficiency carries over to something called few-shot learning as well.
Few-shot, meaning learning from just a handful of examples.
Exactly. It's a measure of how well a model performs when you give very, very few training examples per class, maybe just 4 or 8 or 16 examples.
And how did Threads do there?
It excelled. For example, in a task involving subtyping rare brain tumors, Threads achieved higher accuracy using only 4 samples per class, they call this K4, than its closest competitor, PRISM, managed even when using 16 samples per class, so K16.
Wow, so a four-fold improvement in data efficiency for a rare event prediction. That's huge.
It really is. It means you can potentially build useful models even for extremely rare conditions where collecting large data sets is simply impossible.
And one last point on efficiency, the researchers mentioned Threads is actually physically smaller as an AI model compared to some competitors, right?
Yes, that's another interesting finding. Its slide encoder part is up to 7.5 times smaller in terms of parameters than the encoder used by the GIGAPATH model, for instance.
So smaller but better.
Smaller but better performing, especially on these complex tasks. And the takeaway there is pretty significant for the field. It suggests that a really smart, biologically guided pre-training strategy using diverse multimodal data might be far more effective than just brute forcing things by scaling up the raw size of the AI model. Smarter training beats bigger models, perhaps.
Quality over quantity, in a sense.
Precisely. Now, beyond the standard benchmarks like linear probing or fine-tuning, Threads also introduced some more advanced capabilities that seem potentially revolutionary for clinical workflow, particularly this idea they called molecular prompting.
Molecular prompting. Okay, that sounds intriguing. What is it?
It's a completely novel way to perform what's called zero-shot classification.
Zero shot. Does that mean classifying something the model has literally never seen an example of during its visual training?
Exactly that. Imagine you need to classify a type of cancer slide, maybe based on a newly discovered molecular marker, but you haven't had time yet to collect and label images of that specific marker to train the visual part of the AI.
Okay, how could Threads possibly do that?
It cleverly leverages its molecular branch. Researchers can take known molecular profiles, say the typical DNA or RNA signature of an IDH mutant glioma from a separate established data set, and encode that signature using Threads' dNARNA component.
So you create a sort of molecular template.
Exactly. They call it a molecular prototype or prompt. This molecular prototype acts like a canonical blueprint for that specific biological state, like IDH mutation.
Okay, you have the blueprint, then a new patient slide, a WSI, comes in. How does Threads classify it using this blueprint?
You encode the new WSI using the standard visual encoder part of Threads to get its embedding. Then you simply measure how closely that visual embedding matches the pre-established molecular blueprint prototype you created earlier.
Matches how?
Mathematically, usually using a simple distance measure like L2 distance. Think of it as asking, how close in this learned feature space is the visual appearance of this new sample to the established molecular fingerprint of, say, IDH mutation?
That's incredible. You're basically using stored molecular knowledge to guide the visual classification for a task the visual model wasn't explicitly trained on. Did it work?
It worked remarkably well. They demonstrated this by building prompts for IDH wild type versus IDH mutant status using data from one cancer dataset, like TCGA. Then they tested these prompts instantly on a completely separate, external, unseen dataset.
And the result?
They achieved a very high classification accuracy, measured as AUC, of 0.960, near perfect classification, essentially zero shot, without any task-specific visual training needed for that particular dataset.
Wow. The implications for rapidly deploying classifiers for new biomarkers seem huge. You could potentially test a new molecular marker's visual correlates almost immediately.
It could dramatically accelerate that validation process in the clinic, yeah. Makes the AI instantly adaptable in a way.
And you mentioned the off-the-shelf embeddings were also good for case retrieval, like finding similar past cases.
Absolutely.
Yeah.
Because the universal embeddings that Threads generates are so rich with biological information, thanks to that molecular guidance, they could be used straight out of the box for similarity searches.
So a pathologist sees a really weird or rare tumor.
Yeah, they could feed the slides embedding into a system, and Threads could instantly pull up the most similar cases from a massive database, maybe from TCGA or other cohorts. It significantly outperformed baseline models in identifying similar cancer subtypes in their tests.
Helping with diagnosis confirmation, or maybe even suggesting potential treatment avenues based on similar past cases.
Exactly. AIDS, diagnostic comparison, treatment planning, education, lots of potential utility there, just using the embeddings directly.
Okay, so let's try to wrap this up. What's the big picture takeaway for, the clinician or the researcher listening to this? What does Threads really represent?
Well, I think Threads powerfully demonstrates that the path forward in computational pathology isn't just about getting bigger data sets, but about smarter data integration, specifically multimodal integration.
Using that molecular signal.
Right. By using objective molecular signals to guide how the AI learns visual features from H&E slides, Threads has created this biologically relevant foundation. And the key outcome is that this foundation performs exceptionally well, especially in those data-scarce scenarios.
Lowering the barrier for entry.
Exactly. It
Okay.
significantly lowers the barrier for developing useful AI models for rare diseases, for difficult prognostic questions, for treatment response prediction areas where traditional AI development has really struggled due to lack of data.
And the researchers themselves pointed out something interesting in their analysis about where the benefits are greatest, didn't they?
They did. They noted that just scaling up the size of the pre-training data doesn't seem to provide huge benefits for relatively simple visual tasks like basic tumor grading. The models were already pretty good at that.
Uh-huh.
But they found that for the most complex problems like predicting treatment response or inferring subtle molecular states directly from the image, that's where scaling up the data diversity and especially leveraging that multimodal molecular guidance offered the largest performance gains.
So the complexity of the guidance matches the complexity of the task.
It seems so,
Yeah.
which really leads us to a fascinating question as we look ahead.
Okay, let's hear it. A final thought for our listeners.
Well, given that Threads demonstrates this power of integrating complex, diverse, multimodal data to solve problems where data for the specific outcome is scarce, it makes you wonder, what specific, currently unsolved medical puzzle out there?
Mm-hmm.
Maybe something really complex, like predicting the precise efficacy of combination immunotherapy regimens for an individual patient. Or perhaps identifying minimal residual disease much earlier and more reliably than we can now. What challenge do you think is next in line to benefit most from this type of radically diversified, molecularly guided foundation model? Where can this approach make the biggest difference next?
That is a great question to ponder. Where will this powerful approach take us? That's our deep dive into the world of threads for today. We hope this has given you a clearer picture of how these foundation models are starting to rewrite the rules, potentially, for precision oncology.